later viewing on the 8P YouTube channel. And we also share a link in the 8P Research newsletter that goes out on Substack. So definitely there will be ways to follow up, rewatch, or if you weren't able to watch live, hopefully you can be watching this on the recording. All right, it looks like we've got 17 attendees so far. So that's our usual numbers, around 20. So I think we can get started. So it's my pleasure today to welcome Professor Jason Sheltzer. He is going to be talking to us about a research front that we haven't talked much about, but something we kind of talk about is chromosome therapy. How would you think about ways that you could restoring the normal chromosome balance at chromosome 8? And so we were really excited to hear an update today from Professor Sheltzer. Has a great background, very storied background in this area of chromosome diseases and aneuploidy generally, having trained with Angelica Ammon back in the day. So we're really excited that Jason joined the AP community. So without further ado, I'll pass the mic to you.
All right, thanks so much, Ethan, for the kind introduction. Okay, can people see my slides and hear me okay?
Yes, coming through great.
Great. All right, so like Ethan said, my name is Jason Sheltzer, and I'll talk about some research that we've recently begun using chromosome engineering in aneuploid cells. And if at any point I say a word that people aren't familiar with, if people would like me to repeat anything or go into detail on any points, please feel free to unmute yourself and jump in. And I'm happy to answer any questions you have or do my best to explain anything that we're discussing.
How about this featured image? Where did that come from? Was that your design?
This was a professional illustrator whose name is Julia Cool. Yeah, she generated it as an image of aneuploid cells. You can see the cell has 4 chromosomes, for instance.
Looks great for Halloween as well. Anyway, go
Yep.
for it.
So to give you a bit of background about myself, I am an assistant professor at Yale University. I'm affiliated with the Department of Genetics, the Center for Therapeutic Biology, and the Division of Surgical Oncology. This is my lab over here on the left. And in general, my lab is very interested in using genetic approaches to study cancer and developmental disorders with a particular focus on chromosomal disorders, on syndromes and conditions that are associated with different chromosome imbalances. And for the 8P project, which I'll give an introduction to today, I also want to highlight that the member of my lab who's leading this effort is Sophia Lee, who is both on the call with us and is over here in the photo. So our overall goal is to try to use chromosome engineering to study and potentially in the far off future to think about treating cancer and developmental disorders. So we think about chromosome engineering because I think that chromosome imbalances are very important to a number of different conditions who aren't directly related to one another in terms of cause or even consequence, but they share this theme of being caused by imbalances in different chromosomes. One of the most well-studied examples of a chromosome disorder is Down syndrome, which is caused by having a 3rd copy of chromosome 21, which you can see in this karyotype here. And we think about how you can potentially consider Down syndrome not from the context of individual genes, not from the context of chromosome 21 in particular, but from the context of it is a disorder caused by the triplication of a chromosome. And we want to know if we can revert that triplication, if we can eliminate that 3rd copy of chromosome 21 in this instance, and what we could do how we can accomplish that from a technical perspective, and what we can learn following that. So for Project 8P, we are embarking on a research project to try to generate cells that either have an 8P rearrangement from an 8P hero, and then cells in which that 8P rearrangement has been eliminated, and in which cells have two normal copies of chromosome 8, rather than one copy of a rearranged chromosome 8 and one copy of a normal chromosome 8. And I want to talk for a few minutes about why we think chromosome engineering is a potentially impactful approach to learn more about 8P disorders. So one of the main motivations for this project, using chromosome manipulations and chromosome engineering in cells from 8P heroes, is to generate research tools for further analysis and screening. So you can imagine, for instance, that we have cells that are very kindly and bravely donated from an 8P hero. These are, let's say, stem cells in which one copy of chromosome 8P has a rearrangement, like a deletion or an inversion duplication. We can then take these cells, we can culture them in a petri dish, and then we could differentiate them from stem cells into brain cells or neurons, and we could study how brain cells are formed in cells from an 8P hero. Or we could take cells from an 8P hero, and then we could use them to conduct a drug screen. We could treat them with thousands or tens of thousands of different compounds, and we could look for, just to give one example, a drug that prevents seizure-like activity. And that could be very useful and very therapeutically beneficial, both to learn things about how brain cells are formed in individuals with 8P rearrangements. We could also do chemical screens for any condition that we were interested in trying to treat therapeutically. However, these results would come with a very significant caveat. And so for this experimental setup, for anything that we discovered, for any drug that we discovered was beneficial, or anything we learned about how brain cells were formed, with this experimental setup, it wouldn't be clear whether what we're studying is caused by the chromosome 8P rearrangement, or whether it is caused by each individual's unique genetic background. That is, every one of us out there harbors mutations, and I don't mean mutations in a kind of detrimental or negative sense. Every person harbors unique genetic variation. And if you're studying just one individual cells, any kind of result that you obtain from those experiments could be caused by that genetic variation, independent of the chromosomal rearrangement, which is most interesting and also most pressing to us. So with this experimental setup, you can't rule out the possibility that what you're seeing is caused by each person's genetic individuality or unique genetic background. So instead of setting up experiments like this, we could imagine if we had the capacity to set up a different sort of experiment. Actually, before I go into that, so just one thing to mention is that one way which this problem is commonly addressed is by taking cells both from an affected individual and from their parents. And parents are very closely genetically related to their children. And so having cells derived from parents provides what we'd call a control in these experiments that can somewhat account for each individual's unique genetic variability. But because children are not clones of their parents, parents have unique genetic backgrounds that are not entirely identical to their children, the use of cells from parents is a good but very much imperfect solution to this problem. So another potential solution to this problem would be to use chromosome engineering. So let's say that we had cells from an AP hero, and let's say that we had the technical ability to take those cells from an AP hero, and then we could eliminate the rearranged copy of chromosome 8. So in that circumstance, these cells would be genetically identical. They would be totally the same in all regards. They would have the same mutations, the same genetic background. The only difference would be that one set of cells has a rearranged copy of chromosome 8, and one set of cells does not have a rearranged copy of chromosome 8. We could then take these sets of cells and conduct the same experiments. We could study how each set of cells forms brain cells, or otherwise known as neurons, or we could conduct screens in each set of cells to look for instance, drugs that block seizure activity, drugs that promote correct cardiac rhythms. We could conduct any sort of drug screen here. And now with this experimental design, the only difference between these two cell populations would be the rearranged copy of chromosome 8. And so any difference that we observe or any significant finding from this experimental design, we could then, with more confidence, attribute that difference to the rearranged copy of chromosome 8, which is our motivation, which is what we want to study here. We want to study things that are caused by these rearranged copy of chromosome 8 in our AP heroes. So if we had a way to perform chromosome engineering to start with cells that had a rearranged copy of chromosome 8 and then eliminate that rearranged copy, this could be a very powerful tool for research and for screening. A second motivation for chromosome engineering, for thinking about chromosome 8p not as a collection of individual genes, but as a rearranged chromosome, is that, in my view, single gene explanations for chromosome disorders are very often insufficient to really make tractable progress on thinking about how to address chromosome disorders. And this has been studied, I think that the best we have the most information here from studies on Down syndrome. And to give a brief example from the history of Down syndrome, historically, so Down syndrome is caused by having 3 copies of chromosome 21. And historically, there is an idea that there was a Down syndrome critical region. That is, there was some small region on chromosome 21 where there were one or maybe a few genes whose triplication caused Down syndrome. And the rest of the stuff on chromosome 21 didn't really matter. But what we had to do was we had to map down and identify the one gene or the few genes on chromosome 21, which made up this critical region and which caused Down syndrome. However, as people did more careful studies in individuals with Down syndrome, and they created mouse models of Down syndrome as well, this view was largely overturned. And studies, for instance, from the Reeves lab at Johns Hopkins suggested that there was no chromosome 21 critical region that caused specific Down syndrome phenotypes. And instead of looking just at individual genes as the cause of Down syndrome might be best understood as the sum of the effects of the 200 or so different genes on chromosome 21. So while we are in the infancy of studying chromosome 8p and how 8p rearrangements affect different individuals, by looking at the historical example of Down syndrome, I think we have a clear case of single gene explanations being insufficient to account for the behaviors that are seen in individuals with Down syndrome. And while we are in the very early stages of studying 8p disorders, I think that we have another example of how single gene explanations are insufficient for chromosome 8p as well. And this example comes from the very informative study that was sponsored in part by Project 8p from Wendy K. Chung, where they conducted a clinical and genomic characterization of a large series of 8p heroes. And to highlight one of the conclusions from this study, chromosome 8p contains the GATA4 gene. And GATA4 has been previously linked with congenital heart defects. And we know that a lot of our 8p heroes have various defects in heart formation. However, what this study revealed is that there were structural heart defects in some individuals whose 8p rearrangements did not affect GATA4. And so this suggested that we couldn't learn everything there is about the congenital heart defects in individuals with 8p rearrangements just by looking at this GATA4 gene. So single gene explanations are unlikely to fully account for heart defects in individuals with 8p rearrangements. And maybe we should take a whole chromosome view of chromosome 8 instead to think about this condition. An additional reason why I think chromosome engineering is very important, particularly in the context of chromosome 8p, is that one thing we've also learned is that every 8p hero is unique. To show another image from this previous study from Dr. Chung, this is showing just the wide variety of different inversions, deletions, and duplications that our 8p heroes have. And you can see that every 8p hero is unique. There are, deletions and duplications that are affecting all different parts of chromosome 8p. And so if we were to take a single gene view of 8p, that might only impact a small number of our heroes. And instead, what the similarity here is the rearrangements affecting chromosome 8p. And so maybe if we look at these rearrangements as a whole, rather than looking at single genes, this might be able to shed light on what's affecting a large number of our heroes instead of just a subset who have a deleted region or have a duplicated region in common. So if we think about chromosome 8p as a whole, rather than individual genes, this might open up a kind of broader potential approach. The last point about chromosome engineering that I'd like to make here is my lab is a basic biology lab. We conduct research using cell culture models and using mouse models. That is, we grow cells in petri dishes and we work with mice. We do not run clinical trials, but I think that in the long run, one idea that we're very interested in, and there are many, many hurdles to surmount first, is to think about chromosome engineering from a therapeutic context. The possibility of taking some of this research beyond petri dishes and beyond mice and to think about using it actually therapeutically, though we're very, very far from that right now. And again, I think the most advanced examples here are from the case of Down syndrome caused by trisomy 21, and to show just a few examples of the progress that has been made in Down syndrome using chromosome therapy. This is a paper, it doesn't mention Down syndrome in the title, but it's included in this. So in mouse models of Down syndrome, that is, researchers have previously generated mice that have Down syndrome. Down syndrome in the mouse, mice with Down syndrome are infertile. They're incapable of producing viable offspring. But this research group used a chromosome engineering strategy to restore fertility in mouse models of Down syndrome, which would otherwise be infertile. This is another example of trying to address the defects in hematopoiesis, that is the defects in blood formation, using a chromosome therapy-based approach in cells taken from individuals with Down syndrome. So we are a long way off from that right now, but I do think that chromosome therapy could, in the long run, offer a potentially very powerful therapeutic approach for treating chromosome disorders, including 8p rearrangements. So with that introduction, that's kind of the motivation for what we are, how we're approaching these questions and what we're hoping to achieve for this project that we're working on with the generous support of Project 8p. We are receiving stem cells from different 8p heroes. We are testing and implementing different chromosome engineering strategies on cells grown in Petri dishes to try to remove the rearranged copy of chromosome 8p and replace it with a normal copy without the deletion or duplication or imbalance. And then once we have generated those matched populations of cells that either have the 8p rearrangement or that have two normal copies of chromosome 8, we want to characterize the effects of that 8p rearrangement by studying cell proliferation, by studying gene expression, and by differentiating these cells to form brain cells or neurons, again, in a Petri dish to learn about how the 8p rearrangements are affecting cell biology. And I'm going to tell you about some of what we've done so far and some of what we've planned. And before I dive into that, I just want to give a brief one-slide introduction to a tool that I'm going to mention a lot and that we use a lot in the lab, and that tool is called CRISPR. CRISPR is an approach for targeting specific DNA sequences. Basically, we can use the CRISPR approach to target any region of the genome that we're interested in, including chromosome 8p. And the discovery and characterization of CRISPR was awarded the Nobel Prize in 2020 to Jennifer Doudna and Emmanuelle Charpentier. So CRISPR is something that we use very frequently in the lab, including to target chromosome 8p specifically. So my lab has a long-standing interest in using chromosome engineering to study different chromosomal disorders. And we recently published an approach to use a CRISPR-based technique to perform chromosome engineering. We call this approach REDACT, for restoring disomy in aneuploid cells using CRISPR targeting. And we have a preprint describing this approach on bioRxiv, which is a public manuscript database. So this approach, which I'll describe, allows us to start with aneuploid cells, that is, to start with cells that have a particular chromosome dosage and balance, and then to generate disomic cells from those cells, cells that have two copies of a chromosome rather than three copies of the chromosome. For this approach, we generated a plasmid, a piece of DNA, and this piece of DNA has selection markers on it, which we use for these chromosomal manipulations. First, this piece of DNA has puromycin resistance. This is a positive selection marker. It allows us to select for cells that incorporate this piece of DNA into their genomes. This piece of DNA also has herpes virus thymidine kinase, which is a negative selection marker. And the idea here is that when cells express this negative selection marker, thymidine kinase, or TK for short, they become sensitive to a compound which is normally well tolerated by mammalian cells. So they become sensitive to a compound called ganciclovir, which normally would have no effect in any of us. But if we engineer these cells to express this thymidine kinase gene, then they're killed by the addition of ganciclovir to the culture media. Additionally, this piece of DNA has a fluorescent reporter. That is, it makes cells glow red, which we can use either to find the cells that are glowing red or to eliminate all cells that are glowing red, depending on the purpose of the experiment. So this is a piece of DNA which has a lot of different genetic markers we can use for either positive selection or negative selection. We can then use CRISPR to integrate this piece of DNA onto a chromosome, which we're interested in manipulating. So our scissors here is CRISPR. We're cutting, for instance, on chromosome 1 or chromosome 8p, whatever chromosome we're studying. And then we're integrating this piece of DNA onto that chromosome. Now we kind of have a molecular handle on that chromosome. If we treat cells with ganciclovir, which kills anything that expresses this TK, this thymidine kinase gene, we can then select for chromosome loss. So we can take cells that have, for instance, 3 copies of chromosome 8p, and then we can use this TK construct in order to select for cells that have lost the 3rd copy of chromosome 8p or whatever chromosome we're targeting. And we call this REDACT, restoring disomy in aneuploid cells using CRISPR targeting. So this is an approach to use CRISPR to eliminate a trisomic chromosome or an aneuploid chromosome. And we have applied this approach to generate a number of different chromosomal manipulations. I'll show you some successful instances using cancer cells first. We've been perfecting this technique in cancer cells, which have a number of chromosome copy number changes and are very easy to work with in tissue culture, that is, when growing in Petri dishes. Oh, and before I show that, I also want to just give a brief introduction to how we are identifying chromosomal alterations. We are using a deep sequencing-based approach in order to identify chromosome copy number changes. That is, we take cells, we harvest the DNA from cells, we cut the DNA up into tiny bits, and then we randomly sequence it. And then when we sequence the DNA, we basically count how many pieces of DNA map to each chromosome. If we have more pieces of DNA that map to chromosome 1, that tells us that we have an aneuploidy of chromosome 1 because we have more pieces of DNA to map to it. If we have fewer pieces of DNA from chromosome 2, that tells us that we have lost a copy of chromosome 2 because we have fewer pieces that map to it. And when we do this analysis, when we do this copy number analysis, we generate plots that look like this. So this is telling us the copy number of every region of the chromosome across the genome. This is a cell line, which I'll tell you more about in a minute. You can see that this cell line is diploid for most of the chromosomes. It has 2 copies of chromosome 3, 4, 5, 6, 7, and so on. And then it has a 3rd copy, otherwise known as a trisomy or an aneuploidy of the Q arm of chromosome 1, part of the P arm of chromosome 2, and the Q arm of chromosome 17. So this is the approach that we use for copy number analysis. And I'll show a number of graphs that look like this. And basically just where the red line goes up, that means gain. Where the red line goes down, that means a loss. And if you have any questions about this or want me to explain anything in particular, please feel free to jump in and ask. So we use a DNA sequencing-based approach to identify chromosome gains and chromosome losses. And then we've applied our REDACT technique to eliminate different aneuploidies from different cell lines, focusing first on cancer cells, or I'll show some successful examples from cancer cells. So this is a ovarian cancer cell line. That is, this is an ovarian tumor that was excised from a patient, and then a cancer cell line was derived from it. And you can see that this cell line has trisomy of the Q arm of chromosome 1. That is, it has 3 copies of part of chromosome 1, whereas it has 2 copies of most of the rest of the genome, 2 copies of chromosome 3, 2 copies of chromosome 4, 2 copies of chromosome 5, and so on. Here, I'm just highlighting this trisomy or this aneuploidy of the Q arm of chromosome 1. And you can also see this using a fluorescence-based approach. This is with a FISH probe. This is a probe that lights up whenever it binds to the Q arm of chromosome 1. You can see 3 dots right here. So we used the CRISPR-based chromosome engineering approach that I told you about to eliminate the aneuploidy of chromosome 1q from these cells. So we targeted these cells using the piece of DNA that has the thymidine kinase gene, the negative selection gene. We treated these cells with ganciclovir, which forces the loss of the thymidine kinase gene. And then when we looked at cells following these chromosomal manipulations, we were able to isolate cells that had 2 copies rather than 3 copies of chromosome 1q. So you can see down here, these cells have 2 copies of chromosome 1q. You can see it's flat right here. This is an aneuploidy of chromosome 2, which we did not target. But these cells have 2 copies of chromosome 1q rather than 3 copies of chromosome 1q. So these cells are genetically identical in all regards. They have the same genetic variation, the same set of mutations. The only thing that we did to these cells is we eliminated the 3rd copy of chromosome 1q. This now gives us a very powerful platform to screen these cells or to study these cells and to figure out how this trisomy of chromosome 1q affects the biology of these cells to begin with. Because they have the same genetic variation, the same set of mutations, the only thing that we've done is we manipulated the copy number of a single chromosome arm. We've done this in multiple different cancer cell lines. I'll show you a few more examples of this. This is a stomach cancer cell line. This has a trisomy of chromosome 1q as well. We successfully eliminated a single copy of the trisomic chromosome. This is a melanoma, again, with a trisomy of chromosome 1q. We used the redact approaches that I told you about, the CRISPR-based chromosome engineering approaches. We eliminated the trisomy of chromosome 1q. This is just showing that we can target multiple different chromosomes. This melanoma cell line called A2058, it has multiple different aneuploid chromosomes, and we can target any chromosome in this cell line that we're interested in studying. So here we targeted the chromosome 1q trisomy. You can see this cell line is trisomic for 1q. We eliminated the trisomy. This, the cell line has a trisomy of chromosome 7p. We eliminated the trisomy of chromosome 7p. The cell line also has a trisomy of chromosome 8q in this instance, and we eliminated the trisomy of chromosome 8q. And so we have developed this set of CRISPR-based chromosome engineering tools that allow us to target and eliminate an aneuploid chromosome from cell lines growing in Petri dishes. And we believe this is a very powerful tool that allows us to generate cell lines that we can then study, cell lines that we can do drug screens on, or we can do gene expression profiling, or we can do kind of any experiment we're interested in a situation where the only difference is the altered chromosome, so that anything we discover would then theoretically be due to that altered chromosome. And so I think that this is a very powerful approach that allows us to study different chromosomal aberrations. Additionally, just to highlight one other approach, the 8P research team shared this very nice publication that came out just a few months ago from one of my lab's frequent collaborators, Teresa Davoli at NYU. She published this paper called Karyocreate, CRISPR-based technology to study chromosome-specific aneuploidy by targeting human centromeres. And in their approach, they, again, use CRISPR. They target it to the centromere, or the center part of a chromosome, were chromosomes attached to microtubules, which pull them apart in mitosis. And they were able to basically block the ability of microtubules to bind to centromeres during mitosis to cause targeted chromosome manipulations. And so this represents an additional approach for targeting specific chromosomes for mis-segregation. And I'll talk about this approach a little more in a couple slides. So we have a number of different approaches that we can use to try to use cells donated from an 8p hero and their family, and then in Petri dishes, eliminate the rearranged copy of chromosome 8p to generate cells that have normal copies of chromosome 8, which we can use for research purposes. So our goal is to receive cells donated from 8p heroes and then generate derivatives of those cells in which a normal karyotype has been restored for research purposes. So to dive into the research we've done on this project in particular so far, we've received cells from our first 8p hero as well as from their parents. So this is the karyotype from the parents. And you can see that the parents have normal copies of every chromosome. That is, this red line is pretty flat, showing that there are no chromosomal abnormalities that we can detect in the parent. And then the proband, which is another name for the 8p hero. These are the cells from the proband. And you can see here, this individual has a deletion duplication on chromosome 8p. You can see they have a deleted region here, and then a duplicated region here. And we're very grateful to the individuals who donated these cells for this purpose. So now what we want to do is we want to take these cells and we want to genetically remove this rearranged copy of chromosome 8p and replace it with a normal copy of chromosome 8, which we can then use for drug screening and for cell-based assays. And when we thought about how we wanted to do this, what the approach we wanted to take is we wanted to start with the least invasive approaches. That is, we wanted to use kind of the gentlest manipulations to these cells that we could do first in order to minimize the effect, minimize the possibility that we were causing some other cellular change other than what we wanted. And really the most basic, least invasive approach that we could think of taking is just growing these cells in Petri dishes for a prolonged period of time. That is, not using CRISPR, not really touching these cells in any way, just letting them grow and seeing if over time, just the long period of growth would allow cells that had lost the rearranged chromosome to grow out and to take over the population. And the reason we thought about this is, again, going back to the case of Down syndrome, where this has been most well studied. This has been observed to happen in cells donated from patients with Down syndrome, just when they're grown for prolonged periods of time. So this is one paper reporting that finding in Down syndrome, autonomous trisomic rescue of Down syndrome cells. This is another example from a different aneuploidy condition, Edwards syndrome, caused by a trisomy of chromosome 18. Basically, these researchers took cells donated by individuals with a trisomy of 18. These cells started out with 3 copies of chromosome 18, and then just following prolonged growth in Petri dishes, just if you let these cells grow for some unknown reason, the cells would lose the extra copy of chromosome 18, and they naturally restored a normal 2 copies of chromosome 18. So we thought, this is what's been observed to happen in cells from other aneuploidy conditions. Maybe the same thing, we're just learning about chromosome 8p right now. Maybe we can do the same thing in cells that have these 8p rearrangements. So we took cells very kindly donated by our 8p hero. And then we just grew them in Petri dishes for 40 or 80 days in cell culture. And then we took the cells at the end of either 40 or 80 days, and we asked whether they just spontaneously lost the rearranged copy of chromosome 8. And to look at this, we used a qPCR-based approach. I think the specifics of the technique aren't important, but you can just look here. Cells normally should have 2 copies of every chromosome. We have a probe for the deleted region in the 8p hero and a probe for the duplicated region. So you can see 2 copies of each of these regions in both parents. And then in the 8p hero, starting out, they have 1 copy of part of chromosome 8p, and they have 3 copies of another. So we grew these cells for 40 days, and then we looked at the deleted region and the duplicated region, and there was no change here. They still had 1 copy of this deleted region and 3 copies of this duplicated region. We grew them for an additional 40 days, a total of about 80 days, and things were unchanged. They had a single copy of this deleted region and 3 copies of this duplicated region. And so this suggests that unlike the case of Down syndrome and unlike the case of Edwards syndrome, just growing these cells in culture is not sufficient to cause the loss of the rearranged copy of chromosome 8p. So
James,
that's
possible that
sure.
there are individual clones that might have might have kind of gone through autonomous rescue, but they never somehow outcompeted the others, or they just never rose to detection in this assay.
Right. So I think that's an excellent question. And the idea is that, when we're doing this experiment, we have millions of cells that are growing in a dish, and it could be that this trisomy correction occurred in some cells, but not all cells. And so we'd want to take them out and grow them up and look for that, basically. And I think if we had seen maybe a trend towards the trisomy restoration here, then that was something we were planning to do. Maybe if this blue bar was at 2.8 or 2.7, that would give us some confidence that there was a subpopulation of cells in which this was happening. Unfortunately, that wasn't what our PCR-based approach revealed. And so if it was happening, it would be happening in a rare subpopulation of cells, basically.
That in itself is informative, at least compared to the other chromosome aneuploidies you've looked at.
Exactly. Yep. So this was kind of the least invasive thing we could do, the most basic manipulation. The second approach that we have started to take is to try to accelerate the rate of chromosome mis-segregation with a small molecule inhibitor. So here we have cells in which we are fluorescently visualizing their chromosomes under the microscope. Normally, cells are very careful during chromosome segregation. So these are cells undergoing mitosis, cells going from 1 copy to 2 copies. Here you can see the chromosomes. These are the chromosomes that you can visualize individually. And these cells undergo a normal mitosis, and then they form 2 equivalent daughter cells. My lab works with a small molecule compound called an MPS 1 inhibitor, which screws up this process. Basically, it causes the normally very accurate process of chromosome mis-segregation to go awry. And you can see that here. So here, these are cells that are trying to form 2 equivalent daughters, except you can see that there is basically a chromosome that is stuck between them. That is evidence of a chromosome mis-segregation event. These cells are mis-segregating a chromosome. And so we thought that by treating these cells with this small molecule inhibitor, we could accelerate this rate of loss of the rearranged copy of chromosome 8p. This might be one approach that we could use. And so for this experiment right now, we have the cells donated from our 8p hero. We treated them with this drug to mess up chromosome segregation. And now we are doing what Ethan suggested from the previous experiment. we are growing up cell clones from single cells, and we're going to ask whether they lost the rearranged copy of chromosome 8p or not. And this is kind of the second least invasive strategy that we could come up with to restore the rearranged the normal copy of chromosome 8. In addition, we're starting to implement the other approaches that I've told you about, which we've developed. So we have this approach where we use a CRISPR-based strategy to target chromosome 8 with a negative selection marker with thymidine kinase. We are in the process of starting this approach in the cells from our first 8p hero. And additionally, as I mentioned to you, there was this very nice recent publication from one of our collaborators at NYU, where they target the centromere of chromosomes using CRISPR in order to induce the specific mis-segregation of this chromosome. And the lab that generated this approach has kindly shared the CRISPR, the specific CRISPR sequences that they've used for this. They mailed them to us last month. And this is another approach that we are starting to implement to trigger the targeted mis-segregation of chromosome 8 with an overall goal of generating cells that have two normal copies of chromosome 8 rather than a rearranged copy of chromosome 8. So to sum up what I've told you today, we believe that chromosome engineering is an approach that can be used to generate cell lines in which a rearranged or an aneuploid chromosome is the only genetic difference. We found that prolonged growth, at least for 80 days in Petri dishes, is not sufficient to eliminate the rearranged copy of chromosome 8. We've initiated some additional strategies to target chromosome 8 in cells from our first 8p hero. And then once we have successfully normalized the karyotype in these cells, then we will begin efforts to characterize the effects of these chromosome 8p rearrangements on gene expression and on the production of neurons or brain cells. So the 8p experiments that I've told you about today were done by Sophia Lee, who's a really amazing researcher in the lab. We are extremely grateful for the support of Project 8p and for the 8p heroes who have donated their cells and for the project which is funding these efforts. Thank you so much for your time. I'm really excited to continue this and build on this, and I'd be happy to answer any questions you have about anything I've said today.
Awesome, that was fantastic. If you want to ask a question, please, if possible, come back on camera and either raise your hand or just go for it and ask away. Oh, Nicoletta, please.
Hello, thank you for your great talk. I have lots of questions, but I'll try to perhaps restrict to some. So thank you again for this great talk. So the cells that have these chromosomal rearrangements, they have adapted to live with it. When you remove that, that is putting them out of balance. So have you observed anything that would suggest that, perhaps in the cancer cells or in the non-cancer cells that you are doing transformation for?
Yeah, that's a really interesting question. In the cancer cells, we think that these chromosomal alterations are very tightly tied with the process of malignancy. And so there is a fitness cost when we do these manipulations in cancer cells. However, in cells from individuals with Down syndrome or Edwards syndrome, the opposite seems to be the case. It is believed that in the case of Down syndrome and Edwards syndrome, these cells spontaneously lose the extra chromosome because the extra chromosome is actually detrimental to the cells in some way. The cells are in a more fit, more robust state when they have 2 copies of each chromosome rather than 3 copies of each chromosome. And so in the context of these developmental conditions, it seems like the highest fitness is when the cells have a normal karyotype and they're most robust under those circumstances.
Thank you. And the second question was about the DS-RED as a marker for the cells that have been transformed. Are you happy to keep it for further cell-based assays?
Yeah, so ideally, so this is a question now about the specifics of how we're doing these chromosomal manipulations. And when we're, for this CRISPR-based approach, we are integrating a fluorescent protein onto the aneuploid chromosome. That is, normally our cells do not glow red, but following this genetic manipulation, which we developed, the cells grow red, and that's useful for us to be able to, say, visually identify cells that have picked up this piece of DNA that we're trying to transform them with. However, if we're targeting, for instance, trisomy of chromosome 8, following the ganciclovir selection, the cells will lose the copy of the chromosome that has the fluorescent protein integrated. And so in ideal situation, after our manipulations are finished, when they've lost the aneuploidy, they've lost the fluorescent protein. And so the cells will no longer glow red, and that won't interfere with any drug screening or any type of research that we want to do after the manipulations are done.
Thank you.
Any other questions that I can answer?
Yeah, please. Anyone who has any questions, just either come off camera or at least unmute and feel free to ask or drop your question in the chat. It's another option if you prefer that.
Yep. And, you know, if anything comes up later, any questions you have later, please feel free to reach out to me by email. I shared my email in the chat. Oh, it looks like we have another question.
Jorge.
Hi, sorry, I'm so sorry. I was 10 minutes late to the call. So I feel I normally never ask a question in this circumstance, but as we've got time, I didn't want to miss the opportunity. So I don't think I fully understand. Let me just start with that. But so, because obviously with trisomy 21, you've got 3 copies of, chromosome 21. So it makes sense to me that there would be, if you grow the cells long enough, some will just, spontaneously, like a natural selection, they'll lose that extra chromosome, right? And then they've got the advantage, they'll grow. But with 8p heroes, I just don't understand how that would work the same way, because then, like, they've only got, their one normal 8p, or chromosome 8, and then they've got their chromosome 8 with the anomaly. So if they lose that one, like, then they're still down to 1 copy of chromosome 8. Did I miss the 10 minutes? Are you adding in a chromosome 8?
Yeah, so that's a really fantastic question. And I think that you've accurately highlighted one of the really important differences.
Sorry.
Hello.
Our 8p hero is keen to hear the answer.
Hello. It's one of the really important differences between the examples that I showed you previously from Down syndrome and Edwards syndrome, and I think what will happen in for chromosome 8p. So the first answer is that the monosomic state, the state in which you only have 1 copy of a chromosome, we know is very, very unstable. Cells don't, they will not survive for very long as a monosomy. And in fact, if you have cells as a monosomy, if they only had 1 copy of chromosome 8, something that happens very, very quickly is that cells will reduplicate the single copy of the chromosome, and they will form spontaneously a second copy of the lost chromosome, because the monosomic state is so unfavorable.
Okay. So
In addition,
sorry, go ahead. Yeah.
No, I was just going to say, because you asked about adding extra copies of a chromosome, and this is part of our proposal as well, if that doesn't work. So just to show this, this is a chromosome engineering strategy called microcell-mediated chromosome transfer, where you can directly add an extra chromosome to cells. One of the leaders here is one of our collaborators, Zuzana Storkova. And if our initial attempts to directly target chromosome 8p are not successful, we plan to introduce an intermediate step where we first generate a chromosome 8 trisomy using this microcell-mediated chromosome transfer approach, and then go from the trisomy to the disomy because of the reasons you exactly talked about. So we have a few different possibilities here, and as we continue the research, we'll learn which approach works best.
Gotcha. Okay. Thank you very much.
A couple minutes left here. Any other questions from the audience? If not, I guess one question I had is, we've seen in other trisomies, at least the fibroblasts grown in culture, that there are sort of cellular stresses associated with the aneuploid state, including something called senescence, or sort of like a rapid aging of the cells. How might that, if that's happening in AP, how would that affect your interpretation of the failure to have autonomous rescue so far under 80 days? Is it that, would that confound things? Wouldn't it be selective pressure not to be senescent? Wouldn't a non-senescent cell be healthier than a senescent cell? Or I'm wondering whether there's just more to extract from this result, that you didn't get spontaneous rescue. Because I think it's a sort of a significant result. Even though it's a negative result, it seems like there's something significant to interpret from that.
Yep, I think that's a really good question. So for these cells from the 8p hero, we are growing them as iPS cells, or induced pluripotent stem cells. So these cells have the ability to transform into many different types of cells. We can convert them into heart cells, into brain cells, into lung cells, into any other type of cells. But for now, the cells are maintained in a cocktail of growth factors and drugs and hormones, which try to keep them in the stem state, to keep them in this state where they can become any other cell type. And this cocktail of drugs and growth supplements that they're grown in should prevent them from becoming senescent, because these stem cells, we should be able to grow them in petri dishes forever. And so you're absolutely right that this senescence has been found for other trisomies, but hopefully this won't affect our experiments done in stem cells.
Yeah, so it seems like it's even more significant then that these cells don't have that complicating factor that other cells would have when just growing in the stress of a petri dish environment, and yet they were able to sort of hopefully explore. There should have been some clones in there that could have, had a spontaneous loss of the anomalous AP chromosome, and then they would have filled the hole with the template of the good copy. But if that just didn't have enough of an advantage, then you just would never see it. And so even if it took over even if it was a minority of the population, you might see, as you said, the number, that copy number 3, that ratio might start creeping down, but you didn't even see that. So unless you have to wait even longer periods of time, at some point you just got to call it, right? And say, okay, we didn't see this, so maybe this is not subject to the same sort of selective advantage forces of a typical trisomy. There's, the cells are not freaking out per se, or at least the stem cells aren't. And then it's an open question about other cell types.
Yep. Yeah, absolutely.
Great. We're at the bottom of the hour here. This was a wonderful talk. So I think everyone should be really energized that we're thinking about chromosome engineering. We're thinking about chromosome therapy. We've got a chip on the board there, but it's still relatively early days, and some of the most obvious experiments to try have already kind of yielded a negative result, let's say, but there's still value as we kind of hopefully showed in the discussion. So thanks again, Jason, for a wonderful talk. Thanks everyone for your participation and your usual attendance, because most people are people who show up every month. So thank you for keeping this community solid as we head into the summer. Bina sends her regards and thanks. She's taking a well-deserved little break, so is not able to sort of connect with a strong enough connection, but definitely sends her thanks. So thanks everybody for convening again. We'll join everybody next month. Whitney, who's up next on the roundtable?
It is Dr. Glennis Logston.
Awesome. So yeah, so we'll hear more from the perspective of some genomicists who are going to look at the actual breakpoints, go into the nitty-gritty of the chromosomes and explain how we can actually reconstruct where the chromosome breaks and how how the actual duplication, inversion, a deletion actually occurs from hero to hero. So looking forward to that. Anyway, thank you everybody for joining us. Have a great rest of your week.